Hello guys, I am Dr. Vishnu and today I am coming with an interesting topic. It's regarding drug metabolism. So this actually satisfies your pharmacology syllabus because in the introductory part of your syllabus or anybody who is interested in learning about pharmacology, you know very well that pharmacology is divided into two. One is pharmacokinetics and the other is pharmacodynamics. Pharmacokinetics means what the body does to the drug, and pharmacodynamics means what the drug does to the body. So pharmacokinetics is actually divided into few categories. We have absorption, we have distribution, we have metabolism, and the last is excretion. So today I'm going to talk about drug metabolism. This is also a big presentation, but the number of slides are low, but still a lot of things will be there to explain. So it is divided into two sections. Part 1 will be explained today. And the next part, please do subscribe and stay tuned to our channel. It will definitely come on the next day so that you can view what I want to say. So today we'll talk about drug metabolism and what are the practical things that you need to know when you study drug metabolism. When we come to the mission of our channel, I always understand the struggles of people who study pharmacology. It is not an easy subject. And especially when pharmacology becomes difficult to study, when we enter into a clinical practice or when we study pharmacy practice or when we enter into therapeutics field, it becomes literally very difficult because without pharmacotherapy, therapeutics is also nothing. And when we talk about different books, there are so many books on pharmacology which is available. There are so many notes which will satisfy your syllabus. It is available in apps, it is available in websites, it's available in SlideShare, it's available in various websites. PowerPoints are also available everywhere. In fact, many of my PowerPoints are there in Google SlideShare, where you can go and refer to the slides that I have made on selected topics. But those things only satisfy your exam requirements. Like for example, you have an assignment to submit, you are having a sessional exam, you're having an internal exam, or you are having a university exam. For the syllabus that has been listed out to you, those notes, those PowerPoints may be sufficient. But after that, we have a tendency to forget everything because we studied in the exam perspective. This channel and my personal struggle, my personal attempt is to make you understand how little things that we studied theoretically, we can visualize it in a practical scenario, even though we don't have sufficient hospital exposure or even though we are not in clinical practice. Even then, just by sitting in a place and viewing this video, you can get an insight into a lot of practical scenarios that you can visualize by yourself. So how to interpret bookish notes, PowerPoint information into memory-enhancing practical knowledge, we provide the key. We are here to guide you. We are here with you to make you understand how each and every sentence that you see in the book or literally a lot of things that you see, we can make it in a summary manner and that will be good for your memory and you can visualize it practically. Because I always believe that when you work hard, it is satisfying your academic requirements, but when you work smart, it satisfies both your memory and definitely your professional requirements. So welcome to an ocean of practical knowledge. It's me, Dr. Vishnu, and now we are going to talk about drug metabolism. So what are you expecting? What can you expect from this presentation? There are 9 categories. There will be a general introduction. Then we'll talk about phase 1 metabolism in a summary. Then we have phase 2 metabolism. Then we have enzyme induction and inhibition. Induction means activation. Inhibition means blockade. Drug interactions due to metabolism. How do they occur, etc. Then we will talk about prodrugs, which are the different drugs that are metabolized by cytochrome P450 enzymes. There are some interesting things also. One or two interesting things that I found in one textbook. I thought I will share it with you. And definitely mnemonic time, because mnemonics are something that easily people can remember, some catchy codes, etc. So this is divided into part 1 and part 2. Today I'll be talking about introduction, phase 1, phase 2, enzyme induction and inhibition, and drug interactions due to metabolism. So let's go into the video. Let's go into the talks right away. So always remember, most of the drug metabolizing enzymes are predominantly found in liver. So that is why liver is the primary site for metabolism. And that is exactly the reason why, if you are having very severe liver disease, or you are having chronic liver disease or liver failure, or any kind of condition which is affecting your liver in the long term, then when you are taking some medicines, they may not get metabolized properly. So that can lead to a lot of other issues. So liver is a very important organ in that regards. The enzymes are usually present in endoplasmic reticulum, or they are present in cytoplasm. So if the enzymes are present in endoplasmic reticulum, it is known as microsomal enzymes. And if the enzymes are present in cytoplasm, they are known as non-microsomal enzymes. Drug metabolism usually happens in two phases: phase 1 reactions and phase 2 reactions. So this is just a very brief introduction. Now let's go into the details. When we talk about phase 1, so there are some steps that are written here. So first of all, you are taking a particular drug, it enters into your body. You are taking it orally, it enters into your body. Now when phase 1 metabolism occurs, that breaks down that particular drug that you consume. So for example, you took a particular medicine, it enters into your body. Phase 1 metabolism will break it down, and that step is called drug inactivation. Then a functional group, it can be any kind of functional group. You have studied it, maybe you have studied in chemistry. There are different functional groups. We have aldehyde, we have carboxylic acids, we have alcohol, we have ether. Like that, there are different types of functional groups. So a particular functional group will be added to the inactivated molecule, and that step is known as functionalization. And the function of that functional group is to increase the water solubility of the drug so that it can be properly absorbed. So these are the steps of phase 1. First of all, the drug enters the body. In the phase 1 metabolism, the molecule is broken down. That step is called drug inactivation. A functional group will be added to that inactivated molecule, which is known as functionalization. And then the functional group increases the water solubility of the drug. And this phase 1 metabolism is very, very applicable to all those drugs which are known as prodrugs. Prodrugs are those kind of molecules which are inactive in the first phase. And when they enter into the body and get metabolized, they get converted into the active form. For example, clopidogrel. Clopidogrel is a medicine which is used as an antiplatelet agent. It is a prodrug. Then we have isoniazid, which is a medicine that is used in tuberculosis. So when that enters into the body and gets metabolized, it gets converted into the active form, which shows its anti-tubercular activity. So all those medicines which are prodrugs can have good application in phase 1 metabolism. The metabolism is usually carried out by cytochrome P450 enzymes, and cytochrome P450 enzymes are present in endoplasmic reticulum of the liver and intestinal epithelium. The most common reaction that occurs in phase 1 is known as oxidation. There are different kinds of phase 1 reactions, but the most common that occurs is oxidation reaction. So I am just made for anybody, whoever is related to the medicine field, when they understand or when they read a particular enzyme, what should they understand from it? So I have disintegrated it here. Whenever you hear cytochrome P450, because this is very common. So whenever you hear cytochrome P450, CY means cytochrome, P means the pigment that absorbs light, and 450 is the wavelength at which the particular pigment absorbs light. Cytochrome is a heme protein. and it requires oxygen for drug metabolism. So now you may understand how important oxygen is to our body. So this is how you should understand what the meaning of cytochrome P450. Now, definitely there are a lot of cytochrome enzymes. We have CYP1A2, we have CYP3A4, we have CYP2D6, etc., etc. So what is the meaning of that? So here I have given an example. Whenever you hear an enzyme of cytochrome, for example, CYP1A2. So CY means cytochrome, P means pigment that absorbs light. 1 is the first number that denotes the family. And after that, the alphabet that comes, it denotes the subfamily. And the last number that comes, it denotes the chromosome number. So this is how you can understand for all the cytochrome enzyme. For example, if it is CYP3A4, so 3 is the first number which denotes the family, A is the subfamily, and 4 is the last number which is the chromosome number for the location of a gene for that enzyme. So this is the meaning of cytochrome P450 and definitely the different enzymes of cytochrome. The most common cytochrome P450 enzyme is CYP3A4. Definitely it is very, very common. Many drugs are metabolized through this. And obviously there are other enzymes also. Various enzymes are there. CYP1A2, CYP2B6, CYP2D6, CYP2C19, CYP2C9, and CYP2E1. Apart from medications, enzymes also metabolize various exogenous chemicals that come from environment, food, etc. And that is why you may have heard of the term xenobiotics. XENOBIOTICS, xenobiotics. So these cytochrome enzymes also have an activity to metabolize the different chemicals that come from the environment or from the food that we eat. Cytochrome P450 enzymes also have another function. They synthesize steroids and bile acids. So now we can understand. So if anybody asks you, what are the functions of cytochrome enzyme? So there are 3 functions. Number 1, it helps in metabolizing drugs, the medications that we take. Number 2, it helps in metabolism of different chemicals that usually come from the environment or from the food that we consume. And number 3, they help in the synthesis of steroids and bile acids. Now we come to phase 2 metabolism. So this usually involves conjugation reaction. So all the reactions that are involved in phase 2, their main principle is conjugation. So this usually occurs in non-microsomal conditions, but there is an exception, that is glucuronidation. So glucuronidation usually happens in microsomal conditions. And the most common phase 2 reaction is glucuronidation. So as we said, for phase 1, it is oxidation. For phase 2, it is glucuronidation. But there is a peculiar kind of genetic disorder, you can say, or something, and that is called Crigler-Najjar syndrome. So when you study jaundice, if you are studying jaundice, or especially if you are studying, especially for PharmD students, it will be there in your 4th year therapeutic syllabus, jaundice, or anybody who is studying jaundice, they should know about Crigler-Najjar syndrome because this can lead to jaundice. So Crigler-Najjar syndrome is a condition which is characterized by high levels of bilirubin in the blood. It is severe hyperbilirubinemia. This usually occurs due to the absence of an enzyme that is known as glucuronyl transferase. And when this enzyme is absent, glucuronidation metabolism does not happen. And if glucuronidation metabolism does not happen, you can easily understand what will be the condition of such patients if they take any medicine that is metabolized by glucuronidation. So for example, if glucuronyl transferase is not there in your body, so glucuronidation will not happen. And if you are taking a particular medicine, for example, it is written already here, irinotecan, atazanavir. If you are taking these medicines and you are having Crigler-Najjar syndrome, so these medicines will not get metabolized. So they will stay in your body for long periods which can lead to potential toxicity. They will show their respective side effects. So that is why it is very important to understand a lot of things. So this is a practical knowledge that I gave you. So there are some medicines that are metabolized by glucuronidation. So irinotecan is actually an anti-cancer agent, and atazanavir is a medicine that is used for HIV infection. Now let us look into the different medications that are metabolized by conjugation reactions. So conjugation reactions, there are some examples: glucuronidation, glutathionation, glycination, acetylation, methylation, and sulfation. So through glucuronidation, we can, the drugs that are metabolized are irinotecan. So irinotecan, you know very well, it is an anti-cancer. Atazanavir is a medicine that is used in HIV. Estrogen, you know very well, it is used in oral contraceptive. So you can understand very well what will be the condition of a patient for a female who has Crigler-Najjar syndrome and they are taking oral contraceptive that is high in estrogen. Statins, very interesting. These are used in the treatment of cholesterol and definitely benzodiazepines. So now you can derive the practical knowledge yourself. You first search the side effect of all these medicines, and then you try to understand what will happen if a person with Crigler-Najjar syndrome is taking all these medicines. You can yourself predict what are the different problems that can happen. Then we come to glutathionation. So the drugs metabolized are fosfomycin. Fosfomycin is a antibiotic which is usually given in severe conditions. Doxorubicin, it is an anti-cancer agent. It has a very important side effect of causing cardiomyopathy. Bleomycin is also an anti-cancer agent. It also causes pulmonary toxicity. It's a very significant side effect. Ethacrenic acid is actually a loop diuretic. It is not used nowadays because it has a high risk of causing ototoxicity, that is hearing loss. Then we come to glycination. So it is salicylic acid, nicotinic acid. Then we come to acetylation. This is very interesting because there are some theories which suggest that if a particular drug is metabolized by acetylation reaction, they have a huge risk of causing systemic lupus erythematosus as a side effect. And systemic lupus erythematosus is a condition in which your body's immune system considers your own body as enemy and sends antibodies towards it. But it has not been proved for all the medicines that are metabolized through acetylation. But yes, there are some medicines that are metabolized through acetylation and have been shown to cause SLE as a side effect. So the examples of such medicines are isoniazid, procainamide, sulfonamides, hydralazine. So there is a mnemonic also, it is called SHIP. S stands for sulfonamides, H stands for hydralazine, I stands for isoniazid, and P stands for procainamide. And definitely there are other molecules also, but it has not been proved for them till now. So we have dapsone, which is usually used in leprosy conditions. Then we have nitrazepam. Nitrazepam is not used nowadays. It's a benzodiazepine. Clonazepam. Hydralazine is a very powerful antihypertensive. Acebutolol is a beta blocker. Ambrinon is usually used in heart failure. Amantadine is actually an antiviral, and it is also used in Parkinson's disease. Caffeine derivatives, sulfonamides, they are antibiotics. Procainamide is an antiarrhythmic agent also. And phenelazine is a highly dangerous MAO-A inhibitor because it has a risk of cheese reaction, which I will talk about later when we talk about antidepressants. Then we come to methylation reaction, methyl dopa. So you can remember this very easily. Methylation starts with M, and the drugs that are metabolized by methylation are also starting with M. So it is methyl dopa and mercaptopurine. Methyl dopa was actually the first line choice of antihypertensive during pregnancy, but now it is the second line choice. The first line choice is labetalol. And mercaptopurine, you know very well, it is also known as 6-mercaptopurine. So it is an anti-cancer agent. And through sulfation, usually steroids are metabolized. Now we come to enzyme inducer or inhibitor. So you can understand the terms very well. What is enzyme inducer and what is enzyme inhibitor? So enzyme inducer is a particular molecule which increases the metabolism of other drugs. So for example, you are taking a medicine that is metabolized through CYP3A4 and you are taking another medicine which is an inducer of CYP3A4. So if both medicines are given together, then the metabolism of the natural medicine gets increased. So as a result of which, you can get either supratherapeutic effect or you can get reduced plasma drug concentration. That means you will not get proper effect of that medicine. So you can get supratherapeutic effect also in case of prodrugs, or you may get subtherapeutic effects also. Subtherapeutic means very less effects. So enzyme inducer increases the metabolism of other drugs. So the consequence is subtherapeutic effects. So if you are using an enzyme inducer with another medication that is metabolized by the same enzyme, so you should increase the dose of latter. Latter means you should increase the dose of that medicine whose metabolism is being increased by the enzyme inducer. So the examples of enzyme inducers are rifampin, which is an anti-tubercular agent, phenobarbital, which is a sedative hypnotic and also used as an anti-epileptic agent, phenytoin is an anti-epileptic agent, griseofulvin is an antifungal agent, phenylbutazone is an NSAID, and chloral hydrate. So these are the medicines which induce enzymes. So if you are using other molecules with it, the other molecules dose should be increased. Enzyme inhibitors are also dangerous. Enzyme inhibitor means they will block the metabolism of other drugs. And if other drugs metabolism is blocked, their plasma drug concentration may increase. And in that condition, you have a high risk of toxicity. So examples are ketoconazole. Ketoconazole is an antifungal agent. Cimetidine, it is an antihistaminic, in the treatment of peptic ulcer, acidity. Erythromycin is an antibiotic, macrolide antibiotic, and metronidazole is an anthelmintic, and it is also used as an antibiotic because it has action against anaerobic organisms also. The last condition is very, very significant and it is very interesting also. It is known as porphyrin danger. So if you have studied in biochemistry, you may know what porphyria is. It is a very dangerous disorder. So there is an enzyme called delta ALA synthase. Delta ALA synthase. It is a microsomal enzyme. And this enzyme actually increases porphyrins. So if you are taking phenytoin and phenobarbital, they increase the production of delta ALA synthase. So when ALA synthase increases, that means porphyrins will also increase. So that is why phenytoin and phenobarbital are contraindicated in patients who are suffering from porphyria. Because already in porphyria, ALA synthase is very huge. That is why porphyrins are being produced. And if you are using phenytoin and phenobarbital, they are enzyme inducers. So they will increase ALA synthase. So that is why phenytoin and phenobarbital should never be used in a patient with porphyria. So from here, we can derive a practical knowledge. For example, a patient is suffering from porphyria and they are having epilepsy as a comorbidity also. So obviously, phenytoin and phenobarbital are used as anti-epileptics. So there we should not use phenytoin and phenobarbital. We can switch over to other safer alternatives. So this is regarding enzyme induction and inhibition. Now we come to some very interesting drug interactions that happens due to metabolism. So first we will talk about the role of rifampin. Rifampin is an inducer of CYP3A4. So this is an example. Now, for example, a female is taking oral contraceptive which is rich in estrogen, or if a patient who is suffering from HIV is taking protease inhibitors. And these molecules are metabolized by CYP3A4. So if rifampin is taken with estrogen or protease inhibitors, the metabolism of estrogen and protease inhibitors will increase. So as a result of which it can result in oral contraceptive failure. For example, a female is taking rifampin and estrogen together and she wants to abort the pregnancy. That will not happen because the effect of estrogen will get reduced. And if a patient who is suffering from tuberculosis and HIV at the same time, if that person is taking rifampin and protease inhibitors, so protease inhibitors will not work. So it will increase the risk of resistance. So this is a very important interaction that is seen with rifampin. Then we come to another medicine known as terfenadine. Terfenadine usually causes cardiac arrhythmias. So it has a tendency to cause QT prolongation cardiac arrhythmias. Ketoconazole is an enzyme inhibitor. So ketoconazole blocks the enzyme which metabolizes terfenadine. So when terfenadine and ketoconazole are used together, there is a risk of death or even life-threatening conditions due to cardiac arrhythmias. So this combination also has to be avoided. Anyways, terfenadine is not used nowadays. So that is another topic, but this is just a practical knowledge that I wanted to give. Now, this is a very, very interesting. I believe that through this single slide, you can actually understand a lot of things that is happening in your body. Role of antibiotics. Always remember that rational use of antibiotics means the correct antimicrobial has to be used to the patient based on the correct indication and the correct duration. Because many conditions are happening in which antimicrobial resistance is on the rise. And that is because we use indiscriminately antibiotics, which actually don't have a proper indication. Estrogen is actually metabolized via glucuronidation. So estrogen is attached to glucuronic acid, which becomes water soluble. This water soluble compound is excreted with bile into the intestine. And when it goes into the intestine, there is an area called colon. And in the colon, bacteria produces an enzyme known as glucuronidase. This glucuronidase breaks glucuronic acid. Now the estrogen becomes free and lipid soluble. And this estrogen comes back into the circulation. And this process is known as enterohepatic circulation. So if you are studying pharmacokinetics, you may have come across this term. So once again, let me tell you the chronology of events. Estrogen is metabolized via glucuronidation, and glucuronidation means glucuronic acid is attached to estrogen, and that becomes water soluble. This water soluble compound is excreted with the bile into the intestine, where it goes to the colon. Inside the colon, there are bacteria which release an enzyme known as glucuronidase. This glucuronidase will break the glucuronic acid. And as a result of which the estrogen becomes free and this estrogen comes back into the circulation. Now, if we are using broad spectrum antibiotics, for example, we are using chloramphenicol, we are using tetracyclines or any kind of broad spectrum. Now, what do we mean by broad spectrum? Broad spectrum refers to those antimicrobials which have a variety of, which have action against a variety of organisms. Now, the problem with broad spectrum antibiotic is that they not only kill the infectious organisms, they also kill the beneficial organisms which are there in your stomach. Now, for example, a female does not want to be pregnant and she is taking estrogen and she is taking a broad spectrum antibiotic at the same time. What will happen? Broad spectrum antibiotics will kill the organisms that are present in the colon. So the process that I told you right now, enterohepatic circulation, that will not happen because there is no bacteria in the colon to release the enzyme glucuronidase. So this is a process known as gut sterilization. So estrogen or estrogen related compounds should never be used with broad spectrum antibiotics, or else there is a high risk of oral contraceptive failure. So this is also a very classical example of drug interaction due to metabolism. So that's it regarding this presentation. Next, we will talk about prodrugs in the next part of the video. There are more interesting things that are to follow. I personally believe that you have learned a lot from this. And definitely, if you have any suggestions to make, if you have any feedback, do share with us. If you feel that the informations that we have provided are worthwhile, then do like, share, and subscribe to Maximum People, because this is the era of online learning due to the pandemic or due to many conditions where many people are not getting access to quality education. And through this YouTube channel, you can make your own notes, you can make your own ideas, you can make your own learning at the comfort of your time. So when we are doing these things for you, please do share and subscribe to Maximum People so that practical knowledge awareness reaches to the maximum population. Thank you so much for listening to me, and bye, and see you in the next part of the video.